Hello, everyone. My name is Haniffa, and I am based in Newcastle and also Wellcome Sanger Institute. And I'm going to just very briefly summarize what we've done today, what we've covered today, and also what's in store tomorrow. So we had State of the HCA, and if you look at when we first began in terms of the inaugural meeting at the end of 2016, we've now profiled 47 million cells, 7,000 individuals, 93 publications. What a fantastic achievement. The HCA Research Framework Team Sign also gave us pandemic preparedness and all the work that went into using the reference atlas to find out the viral entry sites and also to understand COVID-19 pathology. And we heard also from both the co-chairs, Aviv Regev and Sarah Teichmann of HCA version 1.0 and 2.0. And 1.0 was defined in the white paper, 100 million cells, 12 organs with some spatial. HCA 2.0, 1 billion cells, all organs, and all singing and dancing with the different technologies. And this is really going to be driven by the biological networks. And you've heard or been introduced to the biological networks through the booths. And I'll come back to the biological networks for our activity tomorrow. We also heard about now is the time that we can begin to answer genetic diversity and also how we can make HCA equitable and also governed by strong ethical principles. In addition, we heard from the panel discussion chaired by John Marioni, how we can query the atlas. A lot of it was lessons and parallels from the Human Genome Project. Do we have one reference? Do we have multiple reference? Do we have an average reference? And we now are in the era where machine learning advances are going to allow us to really begin to pull out the insights from these reference data sets. And we think primarily this is going to be a query of cell by gene, but rightly, it's not just the favorite gene, but a lot of genes that actually tell you about the function of one gene. And how we can define extremes of normality, particularly rare frequency events, transient events, and also diversity. And as the data sets become bigger, lots more individuals being analyzed, we can begin to see how genetic variants play a part with regards to gene expression and how else may we be measuring this in terms of shift in distribution of cell states and function and also tissue architecture. So that was the first two sessions. Then we heard about cross-cutting approaches, beginning with computational or apps really, Azimuth from the Raul Satijas lab, an app to map cells against a reference data set. And then from Peter Kachenko, cell annotation platform for community-driven consensus, really trying to sort of make or empowering the community so that we can actually not just generate the data, but interpret the data together. And then we had David Osumi Sutherland talking about cell ontology, standardization, building on previous work, and how we can, again, come to consensus nomenclature and understanding. Then we heard from Steve Quake of the Single Cell Atlas Zoo, Tabula Miris, Lemur, Drosophila, and also human. We had a session where the attendees could view the different biological network booths, and I hope this is going to make you more excited to learn more about the biological networks, which we'll hear tomorrow. These are networks that are going to drive HCA, allowing atlasing at scale with the right expertise and incorporating diversity. And just finishing with Saren Bowers on GDPR and HCA, how we, HCA Inc. to ensure that we can actually safeguard and allow the data to be used to get the maximum benefit, but making sure that there is trust and communication between all stakeholders involved within HCA. And I want to finish off by saying thank you to all the speakers, chairs, booth guides, and also the HCA ISAC office, who have worked very hard in the background, which you may not be completely aware of. So what's exciting for tomorrow? We've got posters, we've got biological networks showcase and breakouts, we've got a panel discussion on computational approaches and challenges, funding opportunity, and also networking, which unfortunately will have to be virtual in this current world that we live in. So join us tomorrow for 2 poster sessions. The viewing starts much earlier from 7:30 a.m. EDT, and then the second poster session 4 p.m. EDT. And you can also visit the poster at any time during and after the meeting. And if you have a question for any of the presenters, chat or email them during the meeting. And the actual session tomorrow at the main stage begins at 9 a.m. EDT, and we'll kick off with the HCA Biological Network Showcase session. So goodbye from me, and see you all tomorrow.